Hi, how are you, Hannah?
Hello, can you hear me?
Yeah, hi.
Hi, how are you?
Good, how are you?
Good.
Good. How did the exam go? Did you guys get your test scores back or?
We did. We got to see them today after lab.
Ooh, okay, so how did that go?
I did well. I got an A.
Ooh, congrats.
Thank you. But yeah, it was good. I mean, we only brief. She didn't go over it. We just looked at them and then handed them back.
Okay, yeah, that's good. There was the general consensus of the class, like, oh, I did fairly well, like I got a B or an A. There weren't any like, anybody super discouraged or anything.
She said our lab section did like you a lot of people did really like it was like a good curve. Some people did well, some people didn't. And then in her
Okay.
other lab, they either passed or failed. There was no in between.
Oh, okay.
So we had more of an average,
Yeah, more of a bell curve.
yeah, than the other section.
Ooh,
But
and if you
everybody
guys
did,
did that, sorry, go on.
she said everybody did well on the lab and then struggled with the
The
written
lecture.
lecture
Yeah.
stuff.
I think it's you kind of have to figure out, at least for the people who might have struggled, you have to figure out how you're going to study for the lecture portion. Some people know how to do it. Some people have never taken old school lecture stuff. I mean, you've taken 32 at least, if not 33. So like to fill in the blank, but a lot of people don't know how to
Mhm.
take actual like, somebody's talking to you and you have to write it all down.
Mhm.
So I
A lot
mean, a
of
couple
people,
people, sorry.
a lot of people don't go to the live lecture though, I've noticed.
Yes. And
There's
you'll
only like
you'll
15 people.
you'll see that a lot more. I think the lowest I saw it when I was a student was 5. And that
Oh my
included
God,
me and Dr. B.
that's crazy. This is the first Zoom class I've ever taken, and I could not imagine not watching it live.
Mm-hmm.
Like I'd fall behind.
Yeah, and that's what really happens is that people are like, "Oh, I don't want to wake up that early. I'll just watch it later." And then
Mhm.
there's this giant hour and a half video that's sitting in front of them and they don't want to watch it later. So it just kind of builds up and then all of a sudden tomorrow I have a quiz on the material and it's like cram, cram, cram.
Yeah.
But yeah. Well, um, I'll be in class tomorrow for the Tuesday, Thursday peeps. And I don't know if they went over it yesterday, but I just hope that they're not too, discouraged
Yeah.
over their scores. And yeah, the thing is, exam 2 is going to be a lot more information. So
Mhm.
a lot more students tend to struggle with exam 2 over exam 1. Uh, but because of that, she tends to compensate and try to help you guys out a lot. So if you kind of just take advantage of anything she gives you, like if she's going to tell you like, maybe know about all of the antibiotics that you'll learn about, like ingrain that into your brain as hard as possible. But yeah, well, I'm glad that, you know, it was pretty well rounded, at least for Monday, Wednesday. I'm trying to think. But you guys have been doing DNA, right? DNA replication for the last couple of lectures, or the last one.
Yeah, we've only gone through, I want to say like half of chapter 7.
Okay.
Tomorrow's lecture is still DNA.
Yeah. Well, do you have any questions on that front or anything
I
like that?
actually do.
Okay.
Not a lot of questions, but just like 2 or 3.
Yeah, go for it.
Um,
And I'll pull up the
first-
PowerPoint so I can try to also.
Okay. My first question is in chapter 6.
Mhm.
In the study guide, it asked which form of oxygen is most toxic.
Mm-hmm.
And
That's it. I know.
yeah,
that's a
so in the
weird question.
slides, it just tells you what the 4 toxic forms of oxygen are, but I just don't know which one is the most.
Ah, I see. Okay. So usually it's just the radical oxygen that
Okay.
isn't bonded to anything. I want to pull up the 4 so I can get a better idea. Let's see. And this is chapter 6.
6,
Okay.
yeah. And I want to say it's like slide 39, the second to last slide.
All right, let's see. Every time with the Microsoft 365. Okay. Second to last slide.
there,
Okay. Oh no, my batteries are. I should have done this earlier. But singlet oxygen, superoxide radicals, peroxide anion. Okay. Let's see. I want to say that singlet oxygen is the most radical because we have certain enzymes that are able to break down or join forces with the peroxide and the hydroxyl to make them less radical. So the peroxidase mixes with that peroxide anion and makes it just water. Let's see, she didn't have any notes on this at all either. Let me see. Catalase also deals with peroxide. And then the superoxide radical and the hydroxyl radical right here is the superoxide dismutase. That's what it kind of deals with. So the singlet oxygen is just kind of like, I don't know, it's one of those weird ones. Let's see. This talks about the aerobic properties. But yeah.
Well, that's kind of the answer I got too from the context of what she was saying.
Yeah.
I just didn't know why, but I don't know if I need to know why it's the most.
Probably not. If I'm going to say that if it's not explicitly stated on a slide, it's
Okay.
probably not too important. She mostly pulls from the slides, and if she's not going to say it out loud, then it's probably not going to be on the exam.
All right.
But yeah, the whole toxic oxygen thing, it's kind of freaky because we breathe it in, and it's a very key element to us being able to live, but it's also something that can do a lot of damage. So it's like, it's a Freaky Friday.
Yeah.
I know a lot of students, at least during my year, like they could not wrap the idea of oxygen being a radical, or being bad in our body. They couldn't wrap it around their heads for the life of them. I was like, well, I mean, yeah, it's a difficult concept, but you just kind of have to go with it. Do we have to question things? Like, this was when we were like cramming, and I was like, I still don't know what a cephalosporin is. So yeah, but yeah, I would just make sure to kind of know these conversions.
Okay.
And what they do, and then you'll be performing the catalase test in class, so this will make more sense.
All right.
Have you guys, hold on, let me pull out my guide real quick, because have you guys done any labs since?
No, just we started lab 8 today.
Started lab 8. Okay, so that's the only one that you guys have touched base on.
9, I think we started. Oh, we did lab 9 today too.
Oh, okay, so you started that one?
From E. coli and the Dawn dish soap.
Yes, and you
And
guys
then-
separated out its DNA.
Yeah, and then 10 was a take-home lab, so she just went over it. And
Mhm.
as we finish chapter 7 lectures, you'll be able to answer the questions, is what she
Okay.
said.
Yeah. Let's see. Because I mean, I haven't been in lab with you guys, obviously. So I
Mhm.
want to go in Thursday knowing what's happening. So let's see. So did you
We
guys
got-
count the plates yet, or
No, not yet.
that's gonna be fun? If you are in the oh, I don't think Monday, Wednesday, there might be somebody on table 8. Whoever's in table 8 got lucky, because you're gonna see some growth, but you have to count every single colony. So
I don't think we have anybody in table 8.
it might be Tuesday, Thursday, or I don't know if they can join.
Well, she changed our lab or seats today.
That's right. She
Yeah.
changes it after exam, so she might have thrown that out. But somebody's gonna be doing pasteurized milk, so okay.
Yeah. So she adjusted it to whatever tables we had, because I think my new table was 5, but we did the group 4 control or something like that.
Yes. Also, a big word of advice for at least lab number 8. If you go to the last page, there's a question. It says a sample from a local swimming hole is taken by the EPA due to possible contamination from cows in a nearby pasture. Star that and make sure you have that one right. I tell you now because almost everybody, including myself, got that one wrong when we turned in our labs. So if you save yourself a point there.
Okay.
And just try to, if you have a question, ask about it.
Okay.
Let's see. See, look, this is what the fun little tidbits you get. They're all yours for the taking. Let's see. So how are you feeling about like the whole differences between eukaryotic and prokaryotic DNA and the plasmid, that whole concept?
So far, all I have is that bacterial DNA is circular, human is linear, bacterial is in the cytoplasm, and then human is in the nuclear envelope. And those are the only 2 differences I have so far.
Okay. You'll probably get some more tomorrow in lecture and be able to fill the rest out, like she was probably saying. And then this is the take-home lab. Like, I don't know if it's just me, or a lot of people do this. The whole, converting from RNA to amino acid and things like that. I'm dyslexic, so I always have to switch up at least one letter, no matter how many times I look over it. So be careful, take your time, because, it's such an easy thing to do.
So
But, yeah.
I have a question on that one then. So number 1 in that lab 10. So first you copied on the first row, we copied the group number that we were assigned.
Yes.
And then the mRNA was the base, the pairs for the top one. And then tRNA you get from the base pairs of mRNA, right?
So yeah, so
Okay.
you're gonna go from the template DNA, you're gonna do the opposite, right?
Right.
And then from the mRNA to tRNA, you're gonna do the opposite.
Got it. Okay. That's what I was just making sure I did that right. Okay.
You're good, you're good. How I remember it is that, you have your DNA, right? And then you need to put it into an RNA form, and usually that's the codon mRNA. And then there's an anticodon, which is the opposite, against. So you have to flip it again.
Okay, but you have to go in that order: DNA, mRNA, and then tRNA. Like I couldn't go from DNA to tRNA, right?
Well,
That wouldn't.
I'd have to double check my lecture material, but you'll probably get that answer tomorrow. Because there is a specific way that bacteria. Okay, sorry. There is a specific way that bacteria likes to duplicate its DNA. If I can get the words out. Let's see. Aha, I think I found it. Another little word of advice, if you like to draw things out, do it. I'm pretty sure she did this during a lecture, maybe yesterday.
She did. She provided the PDF and said to copy it until you can draw it without seeing.
There you go. That's it.
I will.
So, let's see. Okay, so it goes, you have your template DNA strand. You're going to do your RNA, your tRNA, or I'm sorry, your mRNA. And then you're going to have this template strand of mRNA, and from there you'll have your DP3, the DNA polymerase 3, basically pick up where it leaves off, and then DP1 cuts it out and replaces it with that DNA. Let's see. I honestly want to say that now that I'm looking, maybe it's just been a hot minute since I've looked at this, because it seems kind of useless. Like, why would you have to do tRNA, mRNA if it's just a codon? I'll pull up the slides. I feel bad. I don't know what the answers are.
It's okay. I was just trying to understand, but I mean, I filled it out already. I did it. So
okay.
I just don't know why I had to do tRNA.
Well, tRNA, so that's transfer RNA, right? And then mRNA is my mitochondrial RNA. So if I'm not mistaken, mRNA is the one that goes to the, yes, mRNA is the one that goes to the mitochondrion. And from there, the mitochondrial chromosomes go in with the tRNA segments that are the codons.
Okay.
I hope that makes sense. So the mRNA is the opposite of the codon, right? It's the anticodon. So that's correct. Being fed through the bottom of the ribosome. And when it's fed through the bottom of the ribosome, the segment of the tRNA is going to find the correct meeting point. So remember, there are two opposites. So if we have a circle lined up with like a hole, right? They're going to line up with each other because they're opposites. So that's going to be our anticodon and our codon lining up with each other to make the amino acid. Let me pull it up so that you can I can kind of show you the image that comes in my mind.
Okay.
When I'm talking about that, because I know it's kind of hard to visualize, and I'm pretty sure she touched on this. Yes. Ha, 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 ha. So mRNA is here, right? And it's going to go through our ribosome in our mitochondrion in eukaryotes. In prokaryotes, it just kind of floats, but it still holds the mRNA name. The tRNA right here is what we have. is going to be the opposite of the codon and code for our amino acid. So it's just kind of like filling out the puzzle pieces, I suppose, because we have to make sure that we get an exact copy, right? But we can't just say copy and paste. We have to say, okay, what complements this, right? And the thing that complements what complements it is an exact copy, if that makes sense. So that's kind of what the mRNA, tRNA do. Because if you notice, the template DNA that you get in your scenario, it's basically the same as your tRNA, except all the thymine is uracil.
Correct.
So it's basically a workaround that our body has to do in order to make an exact copy. It gets kind of confusing.
Well, that and like I get to the point where my C's look like G's and I'm like, what is that?
I know. I do the same thing. What I usually do in order to make it look a little bit different is you know how I can just draw it out, I guess. Like if I were to just do a normal C and compared to like a normal G for me, like they look very similar. You probably couldn't tell which one's which. So what I do is I add this little tail on this guy and it helps me see it a little more clearly.
Okay.
I don't know if that's a subconscious thing that you want to work into your handwriting. I know it's always hard to do that.
And I try to go slow so I know.
Yeah, so it's just like, please let me just write perfectly and write perfect little letters so I know the difference. Oh, it's very tedious, especially when you're in an exam and you're getting nervous and you're like, oh my God, I just want to know what it is.
Yeah, I did. I felt super nervous at my exam.
Did you do the lecture lab portion first?
I got everybody rushed in the door the moment she opened it.
Oh,
so
no.
I didn't even have a choice. I was like, okay, I'll just do the lab first.
It's not that bad.
It wasn't, but I felt like I spent a little too much time waiting for people to move from the next station I needed. But I didn't realize that I could have like answered the other parts of the question while I got to that next station.
Okay.
Instead of waiting and then turning the page to the station because there was questions that you didn't have to look at the plates. You just had to say what the difference was between differential and whatever kind of media. So
yeah.
But now I know.
Yeah, now you know. And honestly, that's why I always started with lab first, was because if you think about all the people who start lecture first, they're going to have to transfer over to the lab portion at some moment in time. And that like you thought that it was crowded. Wait until they get over there.
Yeah, because they're going to be way more people on the lecture side.
Yeah, they're going to be waiting on each and every one of those stations, and they're probably not going to have as much time as you did to answer those questions. So
yeah, I took my time, definitely.
But yeah, let's see. Yeah, there some of the stuff for the DNA, at least, it's very much like these specific words you need to know, like semi-conservative. You know, there's certain ones like that you really need to know. Let's see. But the whole like step 1, 2, 3, and 4 with the DNA, the translation step, and then also making sure you know the difference between replication, transcription, and translation. Transcription, translation. I remember sitting there on the exam getting stressed out, and I'm like, which is which, you know? So drilling those in now will help you. Let's see. But yeah,
Okay.
I always thought that DNA and genes were quite cool. That's my backup plan, is if nursing doesn't work out, I'm just gonna become a genealogist or something. I remember doing Punnett squares. I'd always finish first. But yeah. Yeah, so here. This is a good one. So right, DNA transcription to mRNA translation uses the tRNA to make this amino acid sequence. Just things like that. I want to make sure that I said that right. Yeah. Okay. Well, is there any other questions that you might have on this? I know like some of this is like, where are you going and why aren't you stopping the slides? And I'm probably freaking you out.
No, I think I'm good with where we're up to right now. We haven't gotten to like mutations or anything, so
Okay.
so far I think I'm okay.
All right. Let's see.
And drawing, that's my only other fear, is having to draw stuff.
Let me think. I don't think that you'll actually be graded on any drawings, but I think why she drills you being able to draw things so much is because it's going to help you on the exam if she's asking you a question about it, so that you don't make a simple mistake like mixing up transcription versus translation. So that if you draw it out and you draw out the diagrams, you can follow along and answer specific questions that require a little complex thinking. It's kind of like mental math, right? You want to write out the problem so that you don't mix up anything. You don't flip a sign, you don't add instead of subtract. So just things like that is why she probably drills it really hard. I don't think you'll actually be graded on your drawing capabilities.
Okay.
So don't stress it. And like half the guys were like, I can't draw anything. Like, what are you talking about? I'm about to draw. But,
yeah, and like some of the questions in like the study guide and then the labs, it says make sure you can draw, and I'm like, oh, I can trace.
I know, trying to make everything look pretty in my notes, I gave up at a certain point. I was like, it's gonna look like chicken scratch, but it's gonna be there. I can maybe read it, but it's gonna be there. So yeah. Dr. B offered me to teach one of the labs.
Wow.
And I'm kind of, I don't know what to do. I kind of want to do something antimicrobial because I feel like that would kind of, like that one's a pretty okay one to grasp. It's just really interesting. So I don't know, but I might also wait until a little bit because I know exam 2 is going to be a little bit of a more challenging one. So I want you guys to get all the information from her so that I don't accidentally screw anything up.
Lauren didn't tell us.
Yeah, I didn't teach you guys how to do this one thing. And then she puts it on the test for whatever reason. But I think I'll probably wait until exam 3. So yeah, you guys are fine. It's mainly DNA and antimicrobials on exam 2.
Okay.
That's like the most I can remember. I think at a certain point I was just writing on bangs and Advil. So, but I did it. I survived. But yeah. Well, we'll probably have more to review because I'm sure everybody got a chance to rest over the weekend from the exam, but I'm sure you guys are like, we have to go back into this now. So
yeah,
It's gonna, you guys trying to pick up steam again.
Yeah, and I
So-
think we still have like, I want to say maybe like 40 slides left in chapter 7.
Yeah.
That'll still be like 2 more lectures.
You'd be surprised. um because some of the I think most of the chapter 7, the last few, let's see, it's 105 slides. Wow. There's a lot of pictures, I will say, and a lot of videos in the last several slides. So I feel like it'll go a little faster than you think. But it might just take another, it might be like all of tomorrow and then a little bit on Tuesday, and then you'll pick up the next chapter.
So maybe we'll have more questions after tomorrow.
Yeah, I'm almost willing to bet on it because like, you know, you go through high school and you go through all your biologies and you learn how eukaryotic DNA divides, and you're like, this is how DNA is, like this is DNA period. And then you learn about prokaryotic DNA and how it divides and multiplies, and you're like, okay, so everything I knew was wrong or what? And everybody like questions existence for a little bit, but I promise it'll be okay. Let's see. 1, 2, 3, 4. Okay, 5 chapters. And most of it is microbial growth, DNA, and antibiotics, which all just play hand in hand with most of each other. And then I believe, let's see, it's going to be labs 8 through 13, which is 8, 9, 10, 11, 12, 15. I mean, I wouldn't really count last exams, how it was 1 through 7. I wouldn't really say that it's less exam material, but it's less hands-on exam material, if that makes sense. Like, you're not doing the gram stain for exam 2. You're not focusing on microscope, I don't think. Don't quote me on that. For exam 2, you're not doing anything like, we're not learning any new techniques like that. That you have to do under pressure on exam 2. So I feel like that might help.
Yeah.
I don't know if that, I'm just giving you experience, because I know, you know, every exam differs, but yeah. I will say that all the videos on this one, very, very nice.
Okay.
The most of those videos I rewatched and I got it. If I didn't get it the first time, I rewatched and I got it the second time. So
yeah, I'll probably do that.
Yeah. But if you don't have any more questions, if you're good, don't need any study tips. I mean, you obviously got an A, so that was better than what I got on exam 1. I got, I think, an 84 or something like that. Maybe an 86 on exam 2 or exam 1. Because I had to figure out how to study for the class.
Yeah. Well, I didn't know what I was doing either, but whatever I did, it worked.
You estimated correctly.
Yeah, and I'm using every resource she offers, like
Well, there
the
you go.
reviews and the practice, everything. I just did it all.
There you go. And that's how you study for this class.
Yeah. And I also I gave myself a lot of time to study. Like I did a little bit every day.
There you go. And that really helps solidify, because I mean, you know, I don't know if you think about it like this, but like I noticed that at least in like my math classes, if I didn't do math for a week, I forgot how to do any type of math.
Yeah.
So if you don't use it, you lose it, type of thing. But I'm glad that it's working out for you. Keep up the good work. You're obviously doing things correctly.
Yeah. And I'm going to keep asking a million questions.
No, you are good. That is the point. That if you don't understand something, that you ask the questions so that you can gain that knowledge.
Yep.
And like I always say, there's no question that's a dumb question, because somebody else might have it and just be too shy to ask.
Yeah, that used to be me, but not anymore.
There you go. Advocate for your own education. Do it.
Thank you so much for your help.
Yeah, no problem. Have a good night, okay?
You too. Bye-bye.
Bye. I don't think that anybody else is coming, but that's okay. I'll do homework until 8 p.m.